All right, good morning, everybody. Said, let's get everything going. Chat. Okay, so we're on the last leg, stretch, whatever you want to call it, of the course. And unfortunately, it's also one of probably the hardest sections of the course, which is, I'm sure, not what you really want to hear. So essentially what we've been doing, particularly over the last section and a bit, is really kind of building up to kind of this point. And so all of the work that we've been doing on gene expression, right, is really setting up the field for talking about how that's controlled, right? And so it's going to show you the schedule for this one, I do believe, right? So, hey, there's Thanksgiving. Really, there's not a lot left, right? So our final exam is the last day of this month, and it's not a final in the classical sense. It's kind of a final as in the last one, and it will be held in, as we've done it before, on a class period day on the 30th of November, right? So that's day before the last day of classes, same deal as before. And so really we're going to be teaching or learning, depending on, where you're sitting, right up until Thanksgiving. We're going to stuff our faces with delicious food, and you're going to revise for your exam the following week, which kind of sucks. Hopefully it's not going to spoil your Thanksgiving. But then we're done. It's a super weird semester, the way they have the schedule where we go on holiday and then when we come back, we finish. So let me get this out of the way. So basically the bulk of this, and there's a couple of cool topics that I hope we get time to talk about, which is really kind of the application of all that we've learned. And this is stuff that's kind of quite up my alley, so to speak. But the bulk of this is going to be on control of gene expression. Okay, and so really, if you think about it, let's just kind of get back to, if it'll let me. I don't know what's going on with PowerPoint today. No, PowerPoint no working. Give me a second. What are you doing, computer? There we go. No, all right, well, let's just close all those down. Don't know why PowerPoint is having such a flap. Yeah, do those later. Come on. Here we go. So, yeah. This is kind of a sort of a scary looking image. It'll actually make a whole lot of sense once we've been through this. But really, at the end of the day, let's look up some fun pictures. Get out of it. Ah, cool. False colored pictures. Yum. All right, let's oh, tongue bacteria. Tasty. Right, so if you gave yourself good tongue scraping, this is probably some of what you'd get off of there. So these are all the various different, these are some rod bacteria, these are some ball bacteria. I'm not much of a microbiologist, at least not bacteriologist. But And this is soil bacteria. So these are probably mycorrhizae, so some kind of fungi. Anyway, essentially the point I'm trying to make is that these organisms and ourselves, right? If you think about ourselves as individuals in large kind of populations, essentially, they all exist in an environment, right? So soil bacteria here are going to be in actually an extraordinarily harsh environment. Soil is, you might think of it as being like full of bounty and wonder, but it's really a very barren place in terms of life. It's kind of like always think of it as the desert, right? You know, there's oases of life around, you know, kind of cow turds and fallen apples and stuff, but the rest of it's pretty barren. But these bacteria and various microorganisms have to succeed in that environment, right? They have to reproduce, essentially. They have to gain enough nutrients and fight off predators and various other nasties that wish to kill them and reproduce, right? And that's no different from the bacteria that are, pardon me, currently on the surface of your tongue or your skin, right? We have or in your gut, actually kind of, wow, we talk about environments, your gut is a very interesting one, right? So these bacteria have to deal with not only kind of intermittent food supply, right? Because you don't eat all the time, but also other stuff around that also wants that food, right? And I don't know, not so much in your mouth, but certainly in like mucosal surfaces like your nose and your lungs and other places like that, you have to deal with the host immune response as well, right? And so these organisms, simple as they may appear, are actually existing in a very complex environment, right? And that's no different from, oh, let's have a little look. Let's see if we can find some nice pictures. No. I don't know what that is, but I think someone has their mic unmuted. So mute that if you could, whoever has that kicking around. Oh, these are some very, very pretty pictures. So these are confocal. Let's see if I can open that in there. Sorry, I just love looking at microscopy pictures. Can we invigorate that? Yes, we can. All right, so this is an image. These are actually, they look like they're grown in vitro, so it's not exactly what I was after, but it'll do. And so this is a confocal microscopy picture, right, which I think I've shown you laser. So this is from a laser scanning confocal microscope. Would be my guess. It might be other types of funky microscope. But essentially what we have here is a composite image of 3 different colors. One fluorophore, this might be DAPI, a stain which labels nucleic acids, right, particularly DNA. So you can see this is where the nucleus or the nuclei are in blue. We have essentially scanned the image with green, and my guess green is tubulin, right? So the same protein that we talked about in what was it? Cell division. It makes microtubules. They also make these structures which kind of go throughout the cell. And the red, my guess would that be actin, right? Which is another microfilament that's used in kind of cells moving around and stuff like that. So anyway, these cells, if this were a cross-section of your liver, right, their environment too is constantly changing, right? They are born, these cells, right? That's kind of what we call it. These cells are born. They're specified as liver cells because you don't want like a heart cell in the middle of your liver. That's not going to do anybody any good. And then they do their job, which is detoxifying all the various nasties that are either generated by your body or put in your body. right? So ethanol, as in alcohol, is one of those. Ethanol is quite a toxin, or poison, actually, is probably the better word. So anyway, these cells are in a constantly changing environment. They're constantly changing because they may be at different stages of their own individual life, right? And eventually those cells will grow old and die, and they'll be told to kill themselves, right? Through a process called apoptosis. And also the environment around them is constantly changing. You may think of your body as a fairly static environment, but it's really not. It's a very dynamic environment where, you know, things are maintained within a certain range, right? So your body temperature fluctuates within, you know, like a 2-degree range, give or take. Your blood osmolality, you know, how thick your blood is or how watery it is, kind of varies again within a narrow range. Blood glucose concentration, all kinds of stuff, right, are managed within a particular range, right? But there's a lot of movement within that range, and there's a lot of flux around your body. If you just think about what you do of a day, you know, I got up, don't want to get up. At least it's not dark anymore. Had myself some breakfast, got on my bike, rode for like an hour and a quarter, and now I'm sitting on my tush again, drinking a cup of coffee, right? So just in terms of what I'm doing to my body, things are changing a lot from hour to hour, right? And so you think about our cells, our DNA. DNA, I'm not going to say it's static because it's not, as you'll see over the next couple of weeks, but it is a fixed repository of information, by and large, right? Again, always have to be careful making kind of broad statements because there are exceptions to all of this. But that instruction set doesn't particularly change, right, from one moment to the next, right? You know, when I got up this morning, my DNA was what it was. Once I had my breakfast, DNA in all my cells was, you know, the same. You know, while I was riding my bike into work, wondering what crazy nut jobs around me were doing, DNA stays the same, right? But clearly there are very, very different operating environments, very different conditions in which, you know, different cells have different jobs. So while I was busy digesting my breakfast, you know, my gut cells were doing their job. My stomach was producing a bunch of mucus and acid, different enzymes being secreted in my small intestine, things like that, right? So most of my body's kind of interests were all about digesting the food that I just poured down my throat. Yeah, when I started riding my bike, obviously that still had to continue because I can't do without food. But now it's a major shift to generating ATP from glucose and, you know, getting that glucose to where it needed to be, right? So that my muscles could keep generating ATP for contractions. So the DNA stays the same, but what those cells are doing changes, right? And so they're both kind of external signals. You know, the arrival of, you know, a higher concentration of ethanol in your liver, for example, will trigger a particular response. There are internal signals. You know, as ATP gets depleted inside my muscle cells, you know, those muscle cells are going to go, whoa, we need more glucose. All right, come on, chaps, get your act together. And they'll produce more glucose channels to take glucose up from the bloodstream, right? So there's this constant dynamic process. And really, that is at the crux of how you are the way you are, right? How genes are controlled is what makes you into this complex, multi-organ, three-dimensional organism, not organ. It's also what allows bacteria to cope with vastly changing environments, right? Because they don't have the luxury like we do of upping and moving around. See if I can get back to those nice pictures. There we go. So your tongue bacteria, they can't just go, well, tongue sucks, right? I'm going to go to the nose. That seems like a better place. They are, by and large, static, right? They can move around a bit, but they're not going to move super long distances. So they have to change their gene expression to cope with that change in environment, right? But all of this has a cost, right? It costs energy, lots of energy, to express a protein, right? To go through the process of gene expression. You have to make the NTPs for the mRNA, you have to make the RNA polymerases and all the other jazz that transcribes mRNA. Even if you're a bacterium, you then still have to make all of the charged tRNAs, all the ATP and GTP involved in translation, and all the amino acids, right? Unless you're able to get those from your environment. And even if you can, that also has a cost. So gene expression is expensive. So you only want to do it when you absolutely need to, but you don't want to do it any more than you need to, right? It's this kind of tricky balancing act. And that's really what underpins all of what we're going to learn about gene expression over the next couple of weeks. How do we, or how does a cell, all right, and as an organism, obviously we manage that, but really at the end of the day, this is what happens at an individual cellular level, right? So every one of your millions of cells has to do this. Obviously, you know, all your liver cells, they're not going to go, oh, I don't like being a liver cell. I'm going to express genes that are involved in, you know, being a heart cell. You know, that's not going to happen, right? So they still have their distinct identity as a part of a larger organ, but they still are doing this individually at the cellular level, right? And so that's in many ways what makes this topic so complicated, because there are, as I've just kind of explained, or, you know, given you examples of, there are really just a stunning amount of combinations of environments, insults, right? You know, not saying, you fat, to like adipose cell or something. Get it? Adipose fat cell. Anyway, that wasn't a very good joke. My kind of ability to make jokes about molecular biology is a lot less than it is about, you know, other aspects of genetics. You know, but kind of like things that would harm that cell, right? And it's just hugely varied, right? So the actual topic itself is hugely complicated too, right? And so what I want to start with is not this, because this will just scare the monkeys out of you, is some questions and then some just general concepts, all right? And so we're going to build these up and then build on them. And by the end of this, we will be covering some pretty complex stuff, and hopefully you'll be there right with me going, oh yeah, that's totally cool. And really, in many ways, this is, if you want to understand your bodies, you know, and just as a kind of a self-interest kind of deal, this is at the very base of that, right? So even if you studied like the anatomy and physiology, if you're interested in going into medicine fields, what underpins all of those topics in anatomy, in A&P, is control of gene expression, right? And so really this is just, in my mind, it's just a fabulous topic to talk about, because, you know, I just have to poke myself in some random place and there's something to talk about in terms of control of gene expression, right? It's just such a fascinating topic. So we're going to start with some general concepts, because these are, you know, really important to get. And then we're going to go on to prokaryotes, because prokaryotes are, I kind of hesitate to say they're simpler, but they're more constrained in how they control their gene expression. And then we're going to move on to eukaryotes, which is just so many different ways in which eukaryotes control gene expression. So one of those complex formula that has lots of different symbols and ends up with an answer. So there are all kinds of questions that you could ask, right? So what types of cells will a gene be expressed in? This is obviously commonly a eukaryotic gene expression question, right? Because we have multiple different cell types. Could also be a bacterial question. If you think about E. coli, we have bazongas amounts of E. coli all the way through us, on us. I don't know, it's like 1 or 2% of our body weight, or your body weight, is E. coli, right? Alone. About 5 to 6% of your body weight is bacterial, which is actually pretty funky if you think about it. So yeah, E. coli, very benign kind of gut and skin bacteria. It takes in a plasmid from another bacterium, and now suddenly it is pathogenic, causes really nasty urinary tract infections, kidney infections, all kinds of stuff like that, right? And so the difference between that is A, the DNA it has, obviously, but also how that is expressed, right? There's a lot of questions about control of transcription, right? The actual kind of nuts and bolts of gene expression itself, right? Then you've got kind of higher level questions, right? How will you modify that basic process, right? And this is particularly applicable to eukaryotes, which is kind of a foreshadowing of the complexity that we'll come to later. And then also, particularly in eukaryotes, but also in bacteria, how does that gene product, typically a protein, get modified? Where does it go, right? Because the end result of gene expression is a functional product, right? And it's only functional if it has the right shape and it's in the right place. Insulin is a great example of that, right? So all of these questions all come together to give you the sum, like the answer, in a sense, right? What gene is expressed at what level, where, in response to what, and how much. And obviously there's lots of ways in which regulation can occur. If you think about all these different steps in gene expression, all of those steps can be controlled in how well they work or not. So the kind of one that people think about most of all, for good reason, is control of transcription. And so in prokaryotes, generally speaking, not 100%, control gene expression at the level of transcription. By and large, right? Not 100%. They do use translation to do that. So if you remember the whole point I was making about transcription and translation being coupled in bacteria, that's going to be really important later on when we talk about the trip operon. Obviously, RNA processing doesn't apply to prokaryotes because it only happens in eukaryotes, but that's somewhere where a lot of stuff can be modified. And generally speaking, eukaryotes control gene expression at levels all the way from chromatin state, I guess. So U versus heterochromatin. To post-translational, there's another T there. Modification, if I can spell that right, of proteins. Right. And so all of these, and actually there's additional ones before these, essentially in eukaryotes, can apply. Now that's not necessarily saying each gene is going to be applied, controlled by all of these. It will vary. And so it's very much context dependent. Some genes, the expression in terms of functional protein is controlled almost entirely by post-translational modifications. Others, all kinds of different aspects to it, right? It really varies. It's like every single gene has its own unique kind of storybook, essentially, in terms of regulation. And so kind of talking about general concepts, really when we use the language that we use, we're referring to the end result, right? And so now we're going to talk about kind of put in a little bit of language, right? And so typically, well, there's both positive and negative regulation, right? And in terms of what that means, again, think about what is the outcome. And so in negative regulation, you're being negative, right? So you're turning something off. So negative regulation, negative is the outcome. So gene expression is turned off. And this is done almost all, gotta be careful again, right? Making sweeping statements. But much of the control of regulation gene expression is done by proteins binding to DNA, particularly of transcriptional regulation. It's probably a better way of putting it. And so those proteins have a particular sequence that they recognize and a particular shape which they must have to be able to recognize that sequence. Right, and so these, so for negative regulation, those proteins are repressors. So they bind to the DNA and they interfere with transcription. Now, it could be that they do so by preventing things from binding. They could be that they block the RNA polymerase from transcribing the gene, all kinds of different ways in which they can, but they do so by binding DNA. Now, positive regulation, again, think about the outcome. So in this case, the outcome is positive, so gene expression is turned on. And so if it has to be turned on, normally it is off. And so you can think about these really in terms of like switches, right? You know, in an ideal world, I'd wander around my house and at night and the lights would upstairs would be off if no one's up there. Right? So the default state of the lights should be off. And if you need them, you have to turn them on. Right? So it's positive regulation of light switches, I guess. However, when you have kids, as actually Karma's biting me in the backside because I used to do this to my parents, they tend to leave the lights on all the time, right? And so now I have to have some negative regulation and turn them off, right? So I don't waste much money on my electricity bill when there's no one upstairs. Right, so if you think about the outcome, that also tells you what the normal state of play is. And so in negative regulation, gene expression is typically on by default, and it has to be turned off. And this is very much the case in prokaryotes. So therefore, for prokaryotes, most gene regulation is negative. Obviously, again, not going to say all of it because there will be exceptions to that, but by and large, in prokaryotes, most genes are on. And so what that means, in the absence of anything bound to the DNA, a prokaryotic gene will be expressed. Right, so it's kind of like a blank slate, you know, like if there's nothing there, you'll get expression in prokaryotes. So you have to stop it, right? Because obviously, you know, if you're on a hearty supply of glucose, then you don't need to express the operon involved in metabolizing lactose, right? Because glucose is a better food source. And so under those conditions, lac operon has to be turned off. Because to express it would be a waste of resources, right? It's not going to get you anything other than waste energy. Eukaryotes are the opposite by and large. Most genes are off by default. And therefore have to be turned on. Now, there's a bunch of reasons for that. Part of that, the reasons are the default state of chromatin is thought to be repressive, heterochromatin, but also because gene expression in eukaryotes is coordinated by the presence of these things, these transcriptional activated proteins, right, which help turn on transcription. Now, unfortunately, because which we call it had a fit earlier, this one shut down. So if you remember, where are we at? Oh, don't tell me it's not being displayed. Oh, yeah, it is. Oh, to go fiddle farting around. Here we go. And so in eukaryotes, contrast with prokaryotes, all of this stuff here, the RNA polymerase, transcription factors, mediator, tidal box binding protein, et cetera, et cetera, et cetera. All of that can be bound to the promoter and you won't get transcription. I mean, sometimes it will, right? It's, you know, it's not absolute. It's kind of what they call leaky. But most of the times that RNA polymerase will just sit there twiddling its protein thumbs, all right? It won't do anything. There have to be these transcriptional activator proteins bound to the regulatory promoter, this bit here, or the enhancer or something, to turn on transcription. Right, so these transcriptional activator proteins are really important and are actually one of the main ways in which you have tissue-specific gene expression. So that's why your liver, you know, will express alcohol dehydrogenase, right, which is one of the key enzymes in detoxifying ethanol. There's absolutely no point in your eyeballs expressing that, right? Because one really truly hopes you're not going to have a lot of ethanol, you know, kind of sloshing around in your eyeballs, because that would be weird and also very bad. Oh, actually, fun fact. Does anybody know why a lot of kind of blues and jazz singers and players from the 30s were blind? It's because that was the era of prohibition when, you know, the US government banned alcohol, which didn't work very well. And that led to a lot of moonshine, so stills, generating your own alcohol out of, I don't know, potatoes or corn or whatever. And also the rise of the mafia too, but that's kind of like a separate deal. However, if you don't do a very good job, you actually produce some methanol as a byproduct of fermentation. And methanol is extremely toxic. So ethanol is just generally bad for you, although fun as well sometimes. But methanol makes you go blind. And so that's why an awful lot of jazz and blues singers from that era were blind, because they used to drink a lot of moonshine as part of, you know, what they did, and that turned them blind. Actually, you still have that in, I think India actually had a big rash of alcohol poisoning and blindness through similar kind of deal, right? So it's not an old phenomenon, it still happens. Anyway, that's a little bit of kind of like cultural stuff for you. I'm sure there are blues and jazz singers which are blind because they're, you know, congenital defects or something, but that's also why, kind of like a fun fact. Okay, so in terms of kind of pictorially, right? This would be in eukaryotes where we have nothing bound to the regulatory promoter. So even if RNA polymerase assembles there, we don't get transcription, right? And this is kind of the symbol that we use, right? It's an arrow for transcription, and then it's like kind of, instead of an arrowhead, we have a cross. That means no transcription from that promoter, right? And so this protein has to recognize that DNA and bind to it. So two things, right? One, the transcription. Oh, I wish they thought of a different short name. So yeah, this is much more so for eukaryotes, Kiana, that's right. We'll get to prokaryotes in a little bit. So for one, this protein has to be present, right? If it's not present, it's not going to bind to anything, right? Two, the DNA sequence the TAP recognizes, and my apologies for not writing that out before, it gets a bit tedious after a while, has to be present in that gene's promoter. Because even if the protein is present, if the bit of DNA that it can bind to is not, it's not going to bind, is it? So essentially, actually, Kiana's a good question. If you want to flip this on its head and look at it in terms of negative regulation, you just go up instead. And so, actually, can I get a, if this were, sorry, looking for, aha. So if this were, God dang it. Kind of doing this on the fly. So yeah, this might be a little bit confusing, but if this were negative regulation instead, the binding of the repressor, right, would block transcription, and then that repressor being absent would lead to transcription. And so they're essentially the two sides of the same coin. Right, if we're talking about positive, protein has to be bound for it to be turned on. If we're talking about negative, protein has to be absent for transcription to occur. That help? Cool. Note to self, add another slide for negative regulation. Right, I'll have to do that for next semester. Okay, so in terms of negative regulation, now we have kind of an extra couple of wrinkles, right? And so really inducible and repressible transcription apply to negative regulation. And again, I want to be very clear. This is in broad brushstrokes. Negative regulation is prokaryotic and positive regulation is eukaryotic. But there will be exceptions to that, right? You don't find them super often, but they are around. And so just kind of keep in mind that that's a very broad statement. It's not an absolute truth. So again, you have to think about the outcome, right? This is where things get a little tricky because now we have kind of some multiple steps, right? And so essentially, in negative regulation, default is on no proteins bound, right? So if you want to turn it off, now you could have a protein bound to it by default. And that protein then has to be induced to leave, for gene expression to be turned on. And that is called inducible expression. So if you think about this in terms of context, right? Context is everything when it comes to control of gene expression. If you think about, a bacterium, it's going to have, essentially a whole bunch of different tools in its genome to deal with different environments. But one hopes as a bacterium that you don't encounter those weird environments all that often, right? But you have to have the ability to respond to them if you do. And so for all of those kind of like off the wall scenarios or less than ideal kind of situations, you're going to want to have them turned off, like all the time, unless they're needed, right? Because if everything's perfect in your bacterial world, you won't need them.
Professor, for that last part, you're saying that for gene expression to be turned on, it's called inducible expression, or you need inducible expression?
Oh, for gene expression to be turned on, you have to get that repressor to leave. You have to induce it to leave.
Okay.
And so if a repressor is bound by default and that transcription is off, then you have to get that repressor to leave somehow, otherwise you're not going to get transcription. And the thing that does that is called the inducer. And this is often a metabolite, which is, I don't know, the substrate, I guess, of the gene product. So if you have, I don't know, let's say like trehalose, which is a pretty funky sugar. It's often used in cryopreservation, right? So those kind of fish that freeze in the Antarctic, right? They're stuffed full of trehalose. It's a good cryopreservative. Anyway, and normally you wouldn't expect to find that around and about as a bacterium. So any of the genes involved in metabolizing trehalose typically are going to be off, right? And they're going to be off because a repressor is bound to that gene, the promoter, that stops them from being transcribed. Because remember, normally in bacteria, if no proteins are present, that gene will get expressed, right? So if you want to turn them off, you have to physically put something there to stop it from being turned on. I don't know, I guess kind of think about it in terms of like some kind of kiddie block on the light switch, right? Which stops them from turning the lights on. Don't know. Not a very good analogy, but good enough. However, if that bacterium is on, it found itself a frozen fish, right? And it's able to metabolize that trehalose, and there's nothing else for it to eat. There's no glucose around, whatever, it's kind of eat trehalose or die kind of deal. Now you want to express all of those genes involved with metabolizing trehalose because that's your food source. And so typically in this case, trehalose, or the presence of trehalose, would be what induces that repressor to leave, to allow the expression of that gene or genes. right? So inducible transcription, this whole jazz here, really you can think about that as being something that's not needed very often, but when you need it, right? And so when you need it, typically the thing that you're going to be using, whatever metabolite that is, that's going to be what induces that repressor to leave, right? And so essentially you get back to this state up here, if you kind of put your hand over the cross down here, where the DNA is bare and transcription occurs. Now, repressible transcription is the opposite. So in repressible transcription, gene expression is always on, no proteins bound, and has to be turned off by the binding of a repressor. Now, the key here, though, is that that repressor will only bind to the DNA and stop transcription in the presence of a co-repressor. Right. And this is what I mean by that having the correct shape, right? So obviously the DNA sequence has to be present. Otherwise the protein, can't bind to it, but also the protein has to have the correct shape. And so for repressible transcription, in the absence of the co-repressor, the repressor, which is actually called the aporepressor in this case, has the wrong shape and can't bind DNA. So it's kind of like that, let's say. Right, so it doesn't physically fit the DNA. It doesn't recognize the sequence of DNA that it will bind to. Right. Now, when the co-repressor is present, now the protein changes shape. And now it's in the correct conformation to recognize that DNA and bind to it. How does that work for everybody? So there's a lot of language and kind of concepts, but it's really about thinking about what's the outcome and also the context as well. So if you think about repressible transcription, now this is probably something that you're going to need all the time. But when you have too much of it, well, then you can turn it off. So that's a very different context to inducible transcription, which is more about stuff that you don't need very often. But when you do need it, you really have to turn it on. Repressible transcription is more about stuff that you need all the time, but you want to be able to turn it off when you have enough of it. So again, if you think about the context, what was really something quite complex becomes a lot easier to understand. And so if we look at some nice pictures, who doesn't like looking at nice pictures? This is essentially, what I was talking about. So again, they're two sides of the same coin. For inducible transcription, in the absence of the inducer, the repressor has the correct shape. And so will bind DNA and stop transcription. When the inducer is present, now that protein adopts the wrong shape to bind DNA and is inactive. That means it doesn't bind DNA and transcription occurs. Now, if you think about the opposite of that, that's repressible transcription. So now, in the absence of the co-repressor, this protein cannot bind to DNA, and so transcription occurs. Only when that co-repressor is present does that repressor become active and bind DNA, adopts essentially the correct shape. It's all about shape at the end of the day, right? So in inducible transcription, the protein is always in the correct shape, unless the inducer binds, in which case it changes shape and falls off. In repressible transcription, the protein is always in this shape, the wrong one, unless the co-repressor is present, and where it changes shape and binds to DNA.
Are these also showing negative and positive regulation?
This is really more, this is very much about prokaryotes and negative regulation. So inducible and repressible transcription in this context is very much a prokaryotic deal. Because this is really all about repressors and whether they're binding or not. And whether they bind or not depends on the presence or absence of a second factor. And so this is definitely something that is much more about negative regulation. Because if you look in both cases, like the kind of the, I guess, key characteristic, if you want to call it that, is in both cases where transcription is occurring, there is nothing bound to that DNA other than RNA polymerase and stuff, obviously. So here, the promoter is free of other proteins. Here, the promoter is free of other proteins. In both those cases, you get transcription. That help?
Yes, thank you.
Cool. Okay, so we've covered this already. Genes can be regulated in both ways, right? We're going to give you some kind of chuckle, simple examples to begin with, although they feel anything but really it can get quite complex. And also something that's fairly common is what's called autoregulation, right? This is especially common in bacteria, actually. And the idea is that how do you know that you have enough product, right, in terms of metabolite or whatever? Well, that product can also act to regulate its, the transcription of the genes that produce it. So it's like a feedback mechanism, right? Once levels get high enough, they start inhibiting gene expression So you have fewer proteins which generate that product. So it kind of goes back in a loop, essentially. And this is definitely something that we'll be looking at in prokaryotic gene regulation. All right, just before we move on to that, give you a few moments to kind of cogitate, see if anything kind of comes to mind that you need clarifying. Because this is really, really important terminology you really have to understand. Cool. Okay. Well, now we're going to start heading into the weeds, so to speak. And pardon me, we'll probably start on this, but we'll dive deep into it on Thursday. And really, you know, I always sound like a bit of a scratch record or scratch CD, maybe. Doesn't really work the same way. Read ahead. There's a lot of stuff that we'll be covering, and it can be very overwhelming, right? So if you have at least a passing familiarity with some of these terms and concepts, it will sink in a lot better, and more importantly, in a way, you'll be able to figure out what questions to ask, right? Because you're not going to understand it immediately first go, but if you understand it a bit, then it should make it easier to figure out what you don't understand, right? Which will help you a great deal. Okay, so as I said before, in many ways, gene expression is controlled in prokaryotes through the control of transcription, right? And also now we have, actually we covered this before. Let's see. Nope, not that one. What we're talking about. Maybe that in. No. Oh, I swear that was in here somewhere. Oh, that must be in a different PowerPoint, I guess. Doesn't matter. So in prokaryotes, they control or coordinate the regulation of their genes by grouping all of the genes involved in one pathway together, right? And so this is called an operon. And you do occasionally find these in eukaryotes, but it's pretty rare. So in the same process, like as in, I don't know, e.g. a metabolic pathway. They're grouped together into one regulatory unit, I guess, called an operon. So the operon itself is turned on or off. So all of those genes in that operon will be transcribed at the same time or not, right? Depending on control of the transcription of that operon. And all those genes are lumped together into one transcript, mRNA transcript. Called a poly. Here we go. It's kind of bugging me now. I've got to go find that. Where is that? Ah, come on, where the? Oh, here we go. That was the one I was thinking of. Yeah. Finally, at least I found it. And so this is a polycistronic mRNA, right? It's one long mRNA containing, in this case, 3 different genes. And each gene has its own start and stop codon. So it's bookended by begin, finish, translation. And so this one large polycistronic mRNA, pardon me, is translated to generate individual polypeptides. So each polypeptide corresponds to a gene in that polycistronic mRNA. So in many ways, this is a very elegant solution, right? Gene expression in prokaryotes, I actually find quite an elegant process. It's a good word to use, right? It's more complex in eukaryotes, but I think it's more elegant in bacteria. And the reason being is that you only have to control the expression of this mRNA to be able to control the production of these 3 proteins. And if you need all 3 to do a job, then why not just lump them all together? Right? Then you don't have to worry about, oh, I've produced protein A and B, but crap, I don't have C. Right? So I can't metabolize this to the final product that I need because I'm missing one of the things that's really important. And so these are like little toolkits, essentially, if you think about it. Right? We're going to talk about the lacquer operon, which is all about metabolizing lactose. And so all of the things necessary for metabolizing lactose are in the lac operon. So you just have to control expression of the operon, and you either get or don't all of the proteins required for metabolizing lactose. Right. And this is that scary image from the beginning of the PowerPoint. Essentially, in bacteria, this is the general format, right, of a kind of regulatory unit, like a package toolkit, essentially. And you have 2 kind of parts to that. One, you obviously have the operon. So let's type this down because this is important. I'm going to say regulatory unit. I think that's better. Consists of 2 parts. One is the operon being regulated. From which the proteins will be, I don't know, produced, I guess. Not a very elegant language there. And 2, the regulator, which controls the transcription of that operon. So it's a 2-part system. And that regulator is a separate gene, typically nearby, but it doesn't have to be. It could be anywhere in the bacterial genome, with its own promoter, which is essentially always on. So the regulator protein is always produced. At a low to medium level, right? Because you don't need tons of this stuff, but you do need it to be present all the time. And so that protein is going to be free to move around in the cell, in the bacterium, to be able to bind the thing that it regulates, depending on, you know, presence or absence of inducer or co-repressor, depending on how it works. So again, this regulator protein recognizes DNA in the operon that it's going to be regulating. Got a blocked nose, so my speaking is kind of mushy. Now the operon, this consists of, let's blow this up a bit so it's a bit bigger. Come on. That's what I want. Make sure I can actually see what I'm pointing at. So the operon consists of the structural genes, which are, they're not necessarily make structure. What we mean by structural genes are those are the things that encode proteins that do something, right? Let's say they're enzymes in that particular metabolic pathway, right? So that's what we mean by structural genes. And you can have a whole bunch. I think the trip operon has 5 different proteins encoded by that operon. The promoter where RNA pole binds. And this is the key part, the operator. Where the regulator protein binds. And if you look at it, right, so again, remember, prokaryotes are negative regulation. So normally in the absence of other proteins bound, RNA polymerase will transcribe this gene. And so look at where the operator is, right? It is physically between the promoter and the structural genes. So if the regulatory promoter or regulator promoter is bound here, RNA polymerase physically cannot get through. It is blocked. It's like dropping one of those concrete bollard things in the middle of the road. Don't matter how hard you hit it, you're not getting through, right? Unless you don't have your seatbelt on. And so that's key to how this operon is regulated, right? The presence or absence of that regulator protein is essentially what determines whether or not these structural genes get transcribed. Okay, so that's enough from me. We will dive deep into the lac operon. Let's see if I can scare you a little bit into reading ahead. Look at this. What a beauty. All right. So read ahead. It will help. There's a lot of stuff going on there. And the lac operon is a complete little sweetheart compared to the trip operon, which is even worse or more interesting, depending on your perspective. All right, everybody. I have lab in a short while, so I'm going to scoot. And I'll see some of you in a little bit. I'll see the rest of you on Thursday or Wednesday, potentially. See you later, everybody.